Epidermis. Name of this layer of connective tissue.
Lemon appropriate.
Areolar.
That's type. Papillary layer of dermis. Name of this layer.
Reticular.
Reticular layer of dermis. Right, so if it's an epithelium and I ask for type, it's going to be one of the 8, right? Simple or stratified squamous, simple or stratified cuboidal, simple or stratified columnar, PSCC, transitional. It's going to be one of those. Okay? Otherwise, it's a name. If it's connective tissue and its type, it's either the 3 loose, one of the 3 loose, one of the 3 dense from connective tissue proper, one of the 3 cartilages, or bone or blood. Although we don't really focus on bone and blood in chapter 4 because we have chapters that we'll cover later in much more detail. So really it's one of the 9. One of the 3 cartilages, one of the 3 loose connective tissue propers, or one of the 3 dense connective tissue propers. If it's not one of those, then it's a name, right? Those are the types. What does it mean that epithelial cells exhibit polarity?
There's an apical and there's a basal layer.
So they have poles, they have sides that are different. And as far as the names, luminal or apical membrane, and then the rest of it basolateral. Where do you find the cellular extensions, the membrane modifications or specializations? Luminal membrane or basolateral?
Basolateral.
It's always going to be luminal. And then the 2 basic types of modifications or specializations?
Microvilli or cilia.
Right.
Cilia.
Microvilli or cilia. The third possibility is flagellum, but that's specific to sperm. Function of cilia. Move substances across the surface of the cell rather than moving the cell itself, which is what a flagellum does. Microvilli. Increased surface area. Typically for increased transport, to move more stuff. Other characteristics of epithelial cells, epithelial tissue.
Avascular.
What else? Exhibits polarity, avascular. Cellularity, which means?
It's regenerative.
To have cellularity means you're a tissue that
It's connected to other.
is almost all cells. Almost no extracellular matrix. In fact, the only ECM of the epithelial tissue is the basement membrane. That is the ECM. Right? Otherwise, it's just cell, one after the next. That's on the sides, on top. They're all joined together with lots of membrane junctions, leaving little space between them. Another characteristic would be basement membrane. Right? All epithelial cells have a basement membrane. Combination of cilia and mucus. This is in the airways. And together they're moving the trapped particles within the mucus up towards the throat. So what's the name of that mechanism? Mucociliary escalator. So if I ask for the function of PSCC, you're really focusing on the combination of the cilia and the mucus. So the mucus traps particles, and then the cilia moves the mucus with all the trapped stuff into the throat. And then from there, swallowed into the stomach, where the pH is really low and will most likely destroy the microbes. These cells here,
Goblet cells.
what is their product?
Mucin. Secreted via exotitosis when combined with water creates mucus.
Okay.
Very good, Drew.
That's like the one thing I know.
And this layer here. And then he drops the mic and walks out. That's all I know. I'm out of here. So what's the BM here? Basement membrane, not bowel movement. How about this stuff here? It is areolar connective tissue. Type of membrane junction? Tight or occluding here. Gap, the proteins, connexins, this one, desmosome, and this one, hemidesmosome, which connects the basal membrane of the epithelial cell to
The papillary layer of the dermis.
basement membrane and the areolar connective tissue underneath. So the hemidesmosome is helping to attach the epithelial cell to the underlying connective tissue. And those proteins that you see extending down from the hemidesmosome, they go through the basement membrane all the way into the areolar connective tissue. All right, so basement membrane is all of this. What's this layer, top half?
Papillary layer.
Lucida.
Basal lamina. Bottom half. Lamina reticularis or reticular lamina. And then which of those can be subdivided into two parts? Basal lamina, so the top portion is lamina lucida or clear layer, bottom half. Lamina densa or dense layer. Type. Function at this location.
Gas exchange.
Gas exchange.
If we just said diffusion.
Diffusion of what?
Okay, got it.
Here, these are all simple squamous. Here it is lining the heart.
Endothelium.
So it's called endothelium. Here it's forming the serous membrane. So the special name is mesothelium. Right, so you have endothelium on the left, mesothelium on the right. Top right is mesothelium again, just looking over the top from a superior view. Here we have simple squamous forming Bowman's capsule, and then more simple squamous forming the capillary bed. And then the white space there, that's the lumen of Bowman's capsule. That's where the capillary filtrate collects. And then it starts passing through the tubules. And the kidney tubules are primarily made up of what cells? Start with the shape.
Cuboidal.
These are cuboidal cells. And it's one layer.
Simple.
Simple cuboidal epithelium. Why simple and not stratified in this case? Because this is not the duct of a gland. This is a kidney tubule. Why do you want simple and not stratified?
Because its purpose is for absorption and
The purpose is transport. So generally speaking, when it comes to simple squamous, simple cuboidal, simple columnar, right? Or transport. So for simple squamous, diffusion of gases, diffusion of solutes, diffusion of water, which is osmosis, right? Or using hydrostatic pressure to push water across, filtration. Those are all functions of simple squamous. When it's simple cuboidal or columnar, it's typically two things: a combination of absorption and secretion, which is transported in two different directions. In the kidney tubules, technically it's not absorption, because what we are pulling out is actually coming from the internal environment. It was in the blood. Momentarily it became filtrate, but we want to bring it back into the blood, so that's reabsorption. Secretion and reabsorption for the kidney tubules. But if you say secretion and absorption instead of reabsorption, that's fine. Where do you find simple columnar? Digestive tract. What's the function? Secretion and absorption. Right? If it's a simple epithelium, it's going to be involved in transport. This one? Non-keratinized. Stratified squamous. Locations.
Vagina.
Thank you. Start off with the vagina. Oral cavity, esophagus, pharynx, yeah, anus. So entrances and exits. How do you know it's non-keratinized?
Because you can see that.
See the nuclei.
You can see nuclei all the way to the top layer. Top left, bottom. Those are both non-keratinized, stratified squamous. Top right, keratinized. What are these BVs?
Blood vessels.
Blood vessels. What do you call them? Well, the the subpapillary plexus is technically a little bit lower, and it's the vessels that are running parallel to the surface. And then from them, you get the loops.
Yeah.
So this is the subpapillary plexus. These are capillary loops, and they're in the different color maybe. They're in the dermal papillae. This layer here,
Granulosum.
stratum granulosum, and make sure the word stratum is in there on the exam.
When we used the models, it didn't really show, and it's probably a stupid question, but I'm gonna ask it anyway. The dermal papilla, when they extend higher than what we're seeing here, or like on the model, that's on the far right side of the model, that makes the dermal ridge, correct?
So
then on top of that's the epidermal.
Neither of the models actually show dermal ridges, but I know what you're talking about, and I'll draw it here. So we have some images that show this, but I'm pretty sure the models do not. So these are dermal ridges, which is just dermal papilla that go way up, and then come back down and go way up. And then over the top, the epidermis follows that same contour, right? So these are epidermal ridges, these are dermal ridges.
And then the smaller each individual is the dermal papilla.
Yeah.
Got it.
So I'm pretty sure in the model, they keep the layer of papilla pretty uniform straight across, and then they'll put the epidermal ridges over the top, which is, not totally accurate. They should actually show them going up and down. Type. Location. Kidney tubules. Function. Secretion and absorption, or more precisely, secretion and reabsorption. The dark purple line here.
Basement membrane.
How about the white space here?
Lumen.
That is the lumen. What's in there? Capillary filtrate. And anything that we don't pull out of that capillary filtrate is going to be lost as urine. More kidney tubules. Longitudinal section at the bottom, making it very clear that it is simple cuboidal, and the basement membrane is very clear as well. Type?
Stratified cuboidal.
Typically 2, maybe 3 layers. Hard to see the outline of each cell, so you're going to rely on the nuclei. All right, so here's an inner layer of nuclei. You can see 1, 2, 3, 4 of them, and then an outer layer of nuclei. Down here, 2 distinct layers. Location? Some large glands, or more specifically, the ducts of some large glands. And apocrine and eccrine sweat glands are relatively large. This one happens to be which? Eccrine or apocrine?
Apocrine.
That's eccrine.
Oh.
I'll make it bigger. What is it? How do you know it must be eccrine?
Small lumen.
It's got a duct that goes to the surface. Pretty small lumen as well, compared to apocrine. here's your single row of nuclei. Lots of cytoplasm on the other side of it. Location. Digestive tract. All right, so give me 3 locations. Small intestine, large intestine, stomach. Function. Secretion and absorption. What's being secreted? Mucus.
Stratified columnar.
Location. Ducts of some large glands, yet again. So stratified cuboidal, stratified columnar, ducts of some large glands. Like this would be a salivary gland. This is the parotid salivary gland.
PSCC.
Ciliated pseudostratified columnar epithelium, PSCC. There's your cilia, your goblet cells. And together they make a mucociliary escalator. Oh, locations. So respiratory tract, but if I ask for 3 locations, break it up. Trachea, bronchi, larynx, nasal cavity. This one, and this one. Transitional. 2 locations. Bladder and ureters.
It's also in the male urethra.
Proximal urethra, yeah, the very beginning. What's that? Stratified columnar or simple? Stratified, yes. So the urethra has some stratified squamous at the exit, at the external urethral orifice. It's got some stratified columnar, and it's got some transitional. So it starts out with transitional, then tends to become more stratified columnar, and then eventually stratified squamous. So this gland here, thyroid. What type of gland is that? Endocrine. Inside the thyroid, you see these spherical structures called thyroid follicles. Each one has sort of like a shell of cells that separates the inside, called the colloid, from the outside. What type of layer of cells is this? Simple cuboidal. And then why is this a colloid? What must be in there if we're calling it a colloid? Proteins. Happens to be the proteins that store thyroid hormone until it's time to release it. Goblet cell, which is a unicellular exocrine gland, the only one. Inside the vesicles, mucin. How is it released? Exocytosis. So what are we classifying here?
Multicellular
Exocrine glands. Multicellular exocrine glands. Top left, simple tubular. Bottom left, simple alveolar. Top right,
Simple branch.
so simple means the duct does not branch, but this duct branches a lot. Lots of yellow in here.
I thought you were on.
So the branching duct makes it compound, and then is it branched? The secretory units, are they branched? No. So it's compound, tubular. And this one? Compound tubulo-alveolar. Again, no branching to the secretory units. Each one is separated from the next by a duct, by yellow cells. Here, no separation. between each of these secretory units. So it is actually one big secretory unit that branches. So the name?
Branched.
Simple, because it's one duct, branched, alveolar.
At Gmail.
So which one's not shown here?
The simple tubular coil.
Coiled.
Simple coiled tubular. So how are we classifying the exocrine gland?
Modes of secretion.
Based off of how
Mode of secretion, yes. How they secrete. So the one on the left,
exocytosis.
it's called merocrine, uses exocytosis. On the right,
Holocrine.
holocrine.
Exocytosis.
It bursts.
Nope, just cell rupture. Mhm. Example of a merocrine gland.
Sweat.
Both of them, eccrine and apocrine sweat glands. An example of holocrine, sebaceous oil gland. And this mode of secretion, apocrine. And location, mammary gland. All right, what questions do you guys have? So you're going through the question format, right? Practicing answering all those. So it was up to you to create one for chapter 5, but I gave you one for chapter 4. Who hasn't signed the sign-in sheet yet? Because I know there's way more people here than what's on here. So you guys are good, you guys are good, you guys are good, good. We'll do this row and then kind of pass it around that way.
Professor Mark, can you go over the cholecalcifiers, calcitriol? Can you go over that again?
This is George Corpse Grinder Fisher. That's his nickname, Corpse Grinder.
Corpse grinder. Sounds funny.
Yeah. He's a growler.
I can deal with some. I think
You get used to it.
I think the most that I've been able to deal with is I'm gonna have to find it. I know you've heard them.
Yes, I am an encyclopedia of metal. I've heard them all. If I could, I'd teach a class in metal.
Bring me the horizon. That's about as
Yeah, that's
much
like
as I can
we
take.
call that like pop metal.
Really?
It's pretty fluffy.
So I don't know the band, but I heard one black metal, and it was like ears bled,
I love black metal.
eyes bled.
Emperor's my favorite.
It was a lot.
And Behemoth and Montana are really good. I could go on and on. Anything from the questions version of chapter 4? I know we're going to do cholecalciferol in a minute.
Would that be the same as
Oh, you don't need to go.
chapter 5? Because I have a question mark there under synthesis that would be kind of covered
All right,
the
let's do
same.
that.
Okay.
All right, chapter 5, beginning of the study guide, functions of the integument.
Why aren't you up there code?
You guys see a eraser?
Which one?
It's under your plate. It's under your plate.
Oh, okay, same thing.
Aha, I got it.
And I have another one too, the exudate.
Yes. That's so weird because I have a question mark on that one. I have 3 question marks.
Does anyone else need
Yeah. All right, functions, we have synthetic or metabolic. All right, functions for skin. Functions of integument, protection, and then that one we should be more specific. How is it providing protection? Physical,
Chemical, biological.
what else?
Thermoregulating.
Excretion, thermoregulation,
Synthesis. Sensing.
all right? So what is synthesized in the skin?
Vitamin
Inactive vitamin D3 called cholecalciferol. It's modified cholesterol. And if you don't make it in the skin, how else can you get it into your body? Vitamin D fortified foods, dairy, right? Milk. Once you bring it in through your diet or make it in the skin, where does the cholecalciferol go?
Calcitrol.
To be converted into the next intermediate.
Calcitrol.
Goes to the liver, where we form calcidiol. That gets picked up by the kidneys. So cholecalciferol goes into the blood, liver picks it up, makes calcidiol with it, back into the blood, kidneys pick it up and make calcitriol, which is a steroid hormone. And then what is the function of calcitriol? Which we're going to cover in chapter 6 next anyway. It increases absorption of
vitamin D.
calcium from your diet. So what would be the target organ for this hormone in order to absorb more calcium from your diet? Which organ is the absorption organ? Small intestine. Small intestine. So more about calcitriol's function in chapter 6. Physical barrier is pretty straightforward, just the physical presence, something that separates the two environments. What do we mean by chemical barrier?
Infections.
Chemicals on the surface of the skin that are somehow antimicrobial, that protect us against infection. And then there's one other chemical that has nothing to do with immune function,
Melanin.
but it does protect us against something. Melanin. So that's the one chemical that's protecting us from something other than microbes, right? UV damage to the DNA of our keratinocytes. And not just the keratinocytes. When you create this shield of pigments within those keratinocytes, you're also protecting the dermis below. Does that make sense?
Mm-hmm.
You're really protecting everything deep to those keratinocytes. So going back to the immune or defense function of our chemical barrier, what are some of those chemicals and where are they coming from?
Defensin.
Defensins, where's that coming from? Okay. What else? Dermacidin, where's that found?
Sweat.
Which sweat?
Eccrine.
Eccrine sweat? Bactericidal agents found in
Sebum.
sebum. So sweat and sebum both contain a variety of antimicrobial chemicals. Some of them have specific names like defensins, dermacidin, otherwise just as a group, just antibacterial chemicals, bactericidal agents, either way. What else besides these chemicals that inhibit bacterial growth? What else about the surface of the skin? Yes, the acidity. Acid mantle. So what's the average pH of sweat?
3.
4 to 6. Biological barrier?
White blood cells.
Doesn't refer to chemicals, but refers to
White blood
cells. And defense cells are white blood cells. which one is in the epidermis itself? Langerhans? New name? Epidermal dendritic cells. Once you get past the epidermis, and now you're into the papillary layer of the dermis, what sorts of biological barrier will you encounter as a microbe? Macrophages, neutrophils, eosinophils, basophils, mast cells, more dendritic cells. In other words, lots of white blood cells. What's being excreted?
Water, proteins.
So water, but what's the water carrying out in it?
Proteins.
Nitrogenous wastes. Urea, uric acid, for example. How is the cutaneous membrane involved in thermoregulation? It contains eccrine sweat glands, which provide evaporative cooling. What else? It has thermoreceptors, which are required for the negative feedback loops that end up controlling the sweat glands and controlling skeletal muscle, but something else connected to the cutaneous membrane. So you have two of the three effectors: sweat glands, muscles, and cutaneous blood vessels, the cutaneous plexus. Right, so that's a contribution to thermoregulation, the ability to constrict or dilate the cutaneous plexus, right, to increase or decrease heat loss to the external environment. Plus evaporative cooling through increased sweating. What else? So we've got those two. We've got the thermoreceptors. Go ahead and include the hypodermis now. The adipose provides insulation. Hypodermis provides insulation. The cutaneous membrane provides the thermoreceptors, the sweat glands, the cutaneous plexus. Are we missing anything? That's it. Are goosebumps involved in thermoregulation? Do they have a function with thermoregulation?
No.
If we had fur, yes. In furry animals, the erector pili muscles make the hairs stand up, and it creates an insulating layer of air between the skin and the hair itself. But in humans, the hair still stands up, but it's not providing any form of insulation. So goosebumps do nothing, right? That's just the dimpling of the skin from the erector pili muscle contracting. And of course, that does happen when you're cold, right? It's a sign that you're cold, but it's not helping to warm you up or anything.
So when Bill Nye says that your hair does that to try to trap warmer air surrounding your skin, that's completely false.
Right. Maybe scalp hair a little bit, but again, we're talking fur. Maybe we used to have fur. Sensory function.
Meissner corpuscles.
You have a variety of touch receptors. Meissner's corpuscles, Chinian corpuscles, Merkel cells, hair follicle receptor, all provide sensation of touch. It also covers pressure. What else can we sense with the skin, with the receptors in the skin? Pain, temperature, vibration, pressure, which falls under touch. And are these receptors detecting changes in the internal or external environment?
External.
All external. So collectively, all of those receptors can be called exteroceptors. They all respond to stimuli in the external environment. All right. More questions? Huh? Yeah. If it has 4, then that would, I would think, imply synovial membrane, which we didn't get into. So is this from the questions version?
It's from actually the top of the lecture.
Chapter 4?
Yes, sir.
Gotcha. Objective 4. Explain how epithelial and connective tissues combine to form 4 types of membranes and describe the functions of each. So that would be cutaneous, mucous, serous, and synovial. But we don't talk about synovial until chapter 8. So for us, it's the big 3.
Okay.
Yeah. But the 4th is synovial. And you good on the functions of those?
Yeah, I was just, because it said 4, and I'm like, I'm
Wondering what the 4th is?
Exactly.
Yeah. Thank you for bringing that up. I need a textbook.
I have
Just go over them on your own. It's very simple. I don't remember off the top of my head.
It's only like
And I'm not gonna
the
ask you
amount
about
of
it either.
surface area. The amount of surface
No, I was
area.
kidding. It's a surface area thing, and it's how you count them up. And I don't remember what the limbs are worth or what the trunks
Can
were.
I give it a shot?
Yeah, go ahead. I don't know if you're right or not, but go ahead.
Pretty sure I'm right.
1818.
So it's no.
4.5.
So there's an anterior and a posterior percentage, and then there's a overall. So the face is 4.5%. The back of the head's 4.5%. Total is 9.
Okay, so 9's head.
Each limb, upper limb, is 4.5. Combined is 9.
4.5, 4.5. Front, back, 9.
4.5 and 4.5 is 9.
Oh, okay.
together.
Okay.
Each leg is 9, so that's 18.
Like if you can
Your anterior trunk is 18. Your
Hey, there it is.
posterior trunk is 18. Combined 36.
but we don't have to.
And then don't add to
9, 18, 9, 18, and then you have to do the back. Okay. Yeah, this is something that's going to be important if you work in a burn unit, but otherwise, yeah. You can look it up. Do you what? What does it say in the study guide about that? Since we're on that topic, we will go over the different classes of burns. Gosh, is there anything on the study guide about the rule of 9?
But it was on MIT.
So there's page 7.
It was on MIT.
Oh, I see. So under 3rd degree burns, 1A, we talk about the immediate threat to life. Fluid loss estimated from percent of body burned, rule of 9s. So all you need to know about the rule of 9s is it's a way that we can estimate very quickly and crudely estimate percent of body burn. But you don't need to memorize the numbers. You can always look those up. I don't know them off the top of my head, clearly. All right, so burns. Differences. 1st, 2nd, 3rd. How is 2nd different from 1st?
Blistering.
You get blisters on top of inflammation.
Capillary.
Healing is weeks instead of days. Okay. What else?
It's epidermis and part of dermis.
Structurally, the layers affected are different. So 1st degree, it is just epidermis. 2nd,
Epidermis and papillary.
part of the dermis, at least the papillary layer, because what happens if you've affected the entire thickness of the dermis?
And it's a 3rd degree.
Now it's a 3rd degree burn. So 3rd may or may not include hypodermis, but it is a full thickness burn, so that means definitely the entire dermis is damaged. But it may go deeper. Healing time for a 3rd degree.
It's like any skin graft.
So if second degree is a few weeks, I'm sure third is on the order of months and may not repair at all unless we assist with grafting, right? Some sort of wrap, synthetic skin, grafting. So the first 24 hours, the immediate threat to life is too much water loss, dehydration. After that, infection. So check out the Wikipedia link. Look at some of the different types of grafts. I believe an allograft is a graft of the same species. So we can use skin from a human, right? Ourself, preferably, because then we don't have to worry about rejecting the tissue. But you can also get skin from another person, sometimes the cadaver actually. And then I think xenograft is from animal to person, right? Different species. And then we have synthetic skin. Yeah, so check those out. Why can't third degree burns heal on their own the same way second degree burns do? Because you can't form granulation tissue. Why can't you form granulation tissue? Because you don't have stem cells, you don't have blood clot, you don't have blood vessels, right? Everything's destroyed. And it's not necessarily that you don't have any of those things. You don't have enough. You definitely don't have enough. Next question.
Can you go over the transudate, exudate one more time? Or is that not important enough?
Transudate is like serous fluid. So fluid moving across the mesothelium into the cavity between the two layers of the serous membrane, that's transudate. So just think of transferring or transporting across the membrane. Exudate, specifically, it refers to the fluid that escapes the blood vessels during inflammation. Often combined with pus, right, as all the white blood cells come in to take care of the infection. So like the nice cottage cheesy inside of a nasty cyst, that would be exudate. It's fluid, you know, that, and again, all extracellular fluids ultimately come from capillary filtrate, but lots of white blood cells and bacteria, which makes it stinky. Yeah.
So would a demon just be like transudate fluid because it's not exudate, it doesn't have any pus and stuff.
True.
So it's just like serous fluid.
It's interstitial fluid.
Interstitial fluid.
Just too much of it, accumulation. Yeah, so if lymphatics are not removing the excess capillary filtrate as quickly as it's being formed, you end up with edema. But the fluid is just technically interstitial fluid. And then what's another name for interstitial fluid? Relative to connective tissues, the terminology that we use for connective tissues.
Is it ground?
Ground substance. Yeah. The fluid component of the connective tissue, ground substance, that is interstitial fluid. We're talking about the same thing. Any others?
Can we go over the quizzes?
But you have the answers.
Yeah, up here.
The answers to the quizzes?
Yeah.
You have
No.
a-
Yeah, I mean, but can we, as a class, go over some of the quizzes? I know you posted the quizzes.
Yeah. No, is there a particular question, though?
No, I just for practice, because I know that you said that you randomly select questions from the quizzes to pick the exams.
Yeah. But I mean, we could spend hours on those quizzes, so that's why I'm asking, is there a particular quiz or question?
For me, not by anybody else.
Because you guys have the key posted.
What about going over the fluids? I'm having difficulty with, like, you know, we have, you know, these matrix, the ground substance, the interstitial, which I know now are the same, versus like ICF, ECF. Seems like there's a lot of fluids, and I'm
Yeah.
not always sure how they fit into the picture.
Yep.